All right, today we're going to dive into something incredible, the code of life itself. We're talking about the genome, and it's not just some static blueprint. It's more like a dynamic living program that writes the story for every single living thing on this planet, including you. So let's get into it. This quote just nails it, doesn't it? For the longest time, a lot of scientists really struggled with the idea that one simple molecule could hold all the instructions for something as insanely complex as a person. But it does. Every single instruction, it's all packed right inside of you. The best way to think about this is like it's biological software. The scientist Richard Dawkins put it perfectly. He said, the machine code of the genes is uncannily computer-like. It's a code written in just 4 chemical letters, and it tells the proteins, which are the real workhorses of our cells, exactly what to do. So what exactly is this code? Well, to get our heads around it, we need to start with the master copy of all these instructions. The genome, that's the word. It's the complete set of all the genetic material in an organism. Think of it like a giant library. Each chromosome is a huge book, and each gene is a specific chapter in that book. The entire library, all of it, together, that's the genome. For a long, long time, the picture seemed super simple. DNA was this long string, and specific chunks of it, called genes, held the instructions for building specific proteins. One gene, one protein. If you changed the gene, you changed the protein, and you got a different trait. It was simple, it was elegant, and as we're about to find out, it wasn't even close to the whole story. See, as scientists started to actually sequence entire genomes, from tiny bacteria to plants to us, they really expected to find a nice, clear pattern. But what they found instead was, well, it was a lot more surprising. It just seems like common sense, right? A more complex creature, like us, should have more instructions. And more instructions should mean a bigger genome. But does the data actually back that up? And nope, this chart right here just completely demolishes that assumption. As you can see, some plants, even some single-celled amoebas, have genomes that are way, way bigger than ours. So there is no direct link between how big your genome is and how complex you are. This is what scientists call the genome size paradox. And here is the first major clue to solving this whole paradox. Get this. In the entire human genome, less than 2% of our DNA actually codes for proteins. For decades, scientists just dismissed the other 98% as junk DNA. They figured it was just useless evolutionary baggage we were carrying around. So if it's not all genes, what in the world is filling up all that space in our genome? Well, the answer starts by looking not just between the genes, but get this, inside the genes themselves. It turns out our genes aren't these nice, neat, continuous stretches of code. They're interrupted. The parts that actually have the code for the protein are called exons, but in between them are these interrupting, non-coding sequences called introns. The cell has to read the whole thing, snip out all the intron bits, and then stitch the exons together to make the protein. And these introns aren't just tiny little interruptions. They make up about a quarter of our entire DNA. For a lot of our genes, the intron sections are actually way longer than the exon parts that do the real work. But okay. Even if we add up the protein-coding genes and all those introns, we still haven't accounted for most of our genome. The biggest component is something else entirely. This slide right here shows the reality of what we're made of. Look at that tiny little sliver for protein-coding genes. Now look at that absolutely massive slice of the pie, nearly half our genome, that's made of something called transposable elements. This is the real game changer. So what in the world are these transposable elements that make up so much of us? Well, they're often called jumping genes, and believe me, it's for a very good reason. A transposable element, or a transposon, is a sequence of DNA that isn't fixed in one place. It can actually move from one location on a chromosome to a completely different one. The basic way some of these work is a simple cut and paste. The element is snipped out of its original spot in the DNA sequence, and then it gets inserted, pretty much randomly, into a new one. It's like a line of code that can just decide to move itself somewhere else in the program. It's wild. In humans, there are 2 major classes of these jumping genes. You've got lines, which are long, and signs, which are short. And they are absolutely everywhere. We have millions of copies of these things scattered all throughout our DNA. So here's the crucial point. This stuff isn't just filler. When a transposon jumps into the middle of a gene that already exists, it can break it. If it lands in a regulatory region, it can change how a gene is turned on or off. By constantly shuffling the genetic deck, transposons are a massive source of mutation and a really powerful engine for evolution. So we have to completely update our analogy. The genome isn't a fixed, static blueprint at all. It's a dynamic, self-editing, and constantly evolving program. That so-called junk DNA is actually this huge, complex playground for evolution and a layer of regulation that we are only just beginning to understand. Which leaves us with a final, fascinating thought. For decades, we dismissed 98% of our own instruction manual as just junk. Now we know it holds the keys to evolution itself. So what other profound secrets are hiding there in plain sight, just waiting for us to stop calling them junk and finally ask the right questions?